now. And I hope you can hear me, right? Okay, yes, thanks a lot for the invitation and for the nice introduction. So yes, as you can see, and as you've heard already, I will talk about the genomic basis of repeated divergence in marine snails today. And so what I will do is I will actually not focus on like one specific study we have recently done, but I will actually focus on, or I will like give a broad overview of the work on the genomic basis of adaptation that we have done over the past few years in this snail system that I work on, because I think we found out quite a lot of stuff that now forms quite a nice whole story. So I thought it would be a better idea to give you like that whole story more or less. But of course, because I will do that, I will not really just present my own work, but I will present the work of a lot of different people in a lot of different places, mostly at ISTA and Norway University, where I have been working recently, but also, well, that's actually, I'm already lying. So most of the work was probably, to be honest, done at the University of Gothenburg and the University of Sheffield, where many of my collaborators are located. And I would also like to say before I start that if I'm saying something that is completely mysterious, or I'm not telling you what is shown in a plot that I'm showing in the slides, please don't have trouble interrupting me and just asking a question. Yeah, with that, let's start. So yeah, the focus here is on adaptive divergence in a snail system, but for the general overview, I'm showing you a lizard so that you know I'm not just interested in snails. So adaptive divergence, as most of you probably know, happens when you have some kind of ancestral population that then splits into 2 or more daughter populations adapted to different environments. So here, for example, this lizard population split into 2 daughter populations adapted to a green and a gray environment, respectively. And this local adaptation and divergence process can happen in the phase of gene flow. So while the populations are still connected by genetic exchange. And then, of course, it's interesting to know, and it is what many of us are interested in, to understand what makes it possible that we have this divergence between different environments and between different populations while there is still exchange between them. How can they remain separate in the phase of gene flow? And now that for a lot of systems, we have more and more genomic data available, we can address this question using that genomic data. So we can ask what is the genomic basis of divergence and what is the genomic basis of divergence in the phase of gene flow. And what we very generally expect, so I will not go into the theoretical details now, but very generally what we expect is that if divergence happens in the phase of gene flow, that basically genomic divergence is concentrated in specific relatively limited genomic regions, because if selection is relatively strong and focused on a few genomic regions, those regions are better at overcoming the homogenizing effect of gene flow than if we have just a very large number of small effect loci scattered across the genome. So what we, for example, would expect in systems like this is that we have a relatively small number of loci under divergent selection that have large effects on the phenotype and that are under relatively strong selection. We might also expect that loci, if they have small effects, that they are clustered in the genome, so they are very close to each other, so that selection is basically acting on multiple loci together. And we might also expect the role of genomic rearrangements, which I will talk about more later, because they also can lead to a clustering of selected loci in the genome. And so these kind of questions is what I addressed first a couple of years ago in this snail system that I'm working on, and I'm going to introduce that system to you now. So the snail that I'm working on, it's called Littorina saxatilis, and it's a marine or an intertidal system that occurs on rocky shores that looks something like this. And the interesting thing here is that on these rocky shores, as you can see here, we have two quite distinct habitats. So we have cliffs that can be almost vertical and that are exposed to quite strong wave action during the high tide. And on the other hand, we have these quite flat boulder fields where we have a lot of crabs walking around that feed on snails. And those crabs, they cannot walk up the steep cliffs, so that crab predation is limited to these flat boulder fields. And so we clearly have very distinct selection pressures in these two habitats. And we have two snail ecotypes in Littorina saxatilis that have adapted to these two habitats. So we have a wave ecotype on the wave exposed cliffs that is quite small. So it hides actually from the waves in these crevices in the cliff. It also has a large aperture so that it can also have a large foot, and it can attach really well to the rocks when the waves come. And then on the other hand, we have the crab ecotype that lives in the boulder field in this crab habitat, which is much larger than the wave ecotype, has a thicker shell and has a very careful behavior. And all these traits are adaptations to predation. So all this is protection against the crabs. And the important thing to note here is that these snails, obviously, they really occur right next to each other. There's no real spatial distance between them, and these ecotypes are connected by quite a lot of gene flow. So we clearly have a system like this here, where we have adaptation and divergence in the face of gene flow. And so I will talk to you a bit about what happens in these areas where these two different ecotypes are in contact, and how we use these areas to understand the genetic basis of divergence in this system. So here we have a satellite image of one location that we studied. So this is a contact zone between the two ecotypes. We have the crab habitat here, we have the wave habitat here. And what we did is we went there and we sampled snails on a transect along the shore from crab to wave. And you can also see here that this is on a very small spatial scale, because these snails are quite bad dispersers, so they can have quite high differentiation on these very small scales of just a couple of dozens or hundreds of meters. So in this case, what we did is we sampled almost 400 snails along this transect. And then if you look, for example, at the phenotypic change, you can just plot the transect. So here, just the transect along the shore, you plot that on the x-axis, and you plot some phenotypic trait on the y-axis. So in this case, I picked shell length, which is, of course, a measure of the size of the snail. And you can very clearly see here that we have the large snails in the crab area, the small snails in the wave area, as I've already shown in the introduction. And then in between, we have this gradual or clinal change between the two ecotypes, which is a result of the fact that they hybridize in this area where they come together. So we have a hybrid zone here where we have intermediate phenotypes. But as I said, what we are mostly interested in is the genetic basis of divergence in the system. So of course, we also wanted to look at the genetics there. So what we did is we took all these snails and we sampled almost 50, we genotyped almost 50,000 SNPs using a capture sequencing approach. So these are SNPs more or less randomly distributed across the whole genome of Littorina. And then for each of these SNPs separately, we fitted a cline. So this is basically just fitting a model that describes the allele frequency change for that SNP along the shore. So we can make a plot where we have the transect or the distance along the shore on the x-axis, and then we get the allele frequency on the y-axis, again from crab to wave. And we have here, this blue line is just this point where the habitat changes from the crab habitat to the wave habitat. What else did I want to say? Yes, I wanted to say that, of course, I'm just showing a couple of example SNPs here. So I'm showing 10 example SNPs of these 50,000 that we looked at. And you can see for these example SNPs that I'm showing that there is some change in allele frequency between crab and wave, because there is apparently some genetic structure that separates them, which is not surprising. But you can see that these changes are relatively small for those example SNPs, and that is true for most of the SNPs across the genome. But we also find some SNPs that show quite pronounced allele frequency differences. So you can see you go from almost 0 to maybe 0.8 or something like that. So some SNPs change really strongly with the change between the two habitats. And of course, there it is quite likely that these SNPs are under divergent selection and contributing to the differences between the crab and the wave ecotype. And we can also test for this statistically. I'm not going to go into detail now about how we did this because there is no time, but basically we fitted a model that describes the neutral history of the divergence between the two ecotypes. And then we can say what kind of allele frequency change between crab and wave would we expect under neutrality. And we can ask what SNPs are not consistent with this neutral model of allele frequency change between crab and wave. And those SNPs are then likely to be under divergent selection. And those are actually those SNPs colored in red here. And we call them outlier SNPs, analogously to what people do when they do genome scans, for example. So outlier SNPs are always the SNPs where we have evidence that they are affected by divergent selection between the crab and the wave ecotype. So we find quite a large number of such SNPs. And then, of course, we wanted to know where are these SNPs in the genome. So what I'm showing you here is the Littorina saxatilis genome. So we just have 17 linkage groups or chromosomes that are indicated by these gray bars here. And then in red, I am showing you where these outlier SNPs that we found are located in the genome. And so you have these red points, and the redder it is, the more outliers are there at that position. So we don't have a perfect resolution. So you often have multiple SNPs overlapping. So the darker these points are, the more SNPs you have at that specific position in the genome. And what you can see here quite clearly, I think, is that you have some SNPs like scattered more or less across the genome, but you definitely have very clear clustering of outlier SNPs or selected SNPs in particular regions of the genome. So particularly here, linkage group 6, linkage group 14, and linkage group 17, you find these clusters of outliers. And then, of course, you might wonder, okay, what is there in these regions? Why do you have these big clusters? And actually, in a separate study, we found that in some of the, or in all of these very interesting regions, there is a presence of a chromosomal rearrangement, a chromosomal inversion at that position. So here with these turquoise bars, I'm showing you the location of chromosomal inversions in the Littorina saxatilis genome. And you can see that they perfectly coincide with this block of outliers on chromosome 6 and also chromosome 17. And also in linkage group 14, we have at least an overlap between this cluster and a large chromosomal inversion. So of course, we thought this is quite interesting. And so I'm going to talk a bit more about this pattern. So what is going on here? So basically what is happening here is that where we have an inversion, there are two possible arrangements of the genome. So the genome can be normal. So I call this a standard arrangement where you have some order of genes that I'm calling ABC here. But then there's also an alternative version of the chromosome where you have that inversion, where basically the genome is flipped the other way around and the order of all the genes is inverted. So you have these two possible versions of the genome that both exist in the species. And the important thing is that if you have these two different arrangements, there is no recombination possible between them because the chromosomes cannot align properly during meiosis. So this means that differences between these regions, they can just freely accumulate and will not be homogenized by recombination and gene flow. So this arrangement will get more and more different from this one at all or not all, but many different SNP positions within that region of the genome. And of course, if you have these two blocks that are very different, if one of them is associated with the crab ecotype and the other arrangement is associated with the wave ecotype, then you will have very high differentiation in that region of the genome, and you will find a lot of outliers in that region of the genome. And that is exactly what is happening here, for example, in this linkage group 6 cluster of outliers that I showed you. So we have one arrangement in crab and the other arrangement in wave. And so why is it that these regions that are potentially under selection are coinciding with these chromosomal inversions? This is probably not a coincidence if we find so many examples of this. So, and this is actually explained very well by theory that already came out quite a long time ago, but for which the empirical evidence is mostly much more recent. So to understand why we would expect inversions to contribute to local adaptation in such a system, we can look at this relatively simple schematic where we are comparing a region of the genome where we have a chromosomal inversion and a region of the chromosome where everything is normal. So that is just called the collinear part of the chromosome. And we have this chromosome in one environment and the other chromosome in another environment, and the two environments are connected by gene flow. And then importantly, within these chromosomes, we have these different loci which have locally adapted alleles. So we have the light green alleles in the light green environment, the dark green alleles in the dark green environment, because we have local adaptation at all these loci. And then what is important is when we have gene flow, we of course get mixed genotypes because of gene flow and recombination between the chromosomes from these different environments. And then if we first look at the region of the chromosome where we don't have an inversion, we see we get these mixed up genotypes where we have some alleles that are adapted to this environment and we have some alleles that are adapted to that environment. So we have all these genotypes which basically don't work properly in either environment. So it's not a good idea to mix up stuff like that in a system with gene flow. But then on the other hand, if we look at the inversion, as I told you before, recombination is not possible or it's very, very weak in these regions containing inversions. And this means that these blocks of locally adapted alleles, they cannot be mixed up. So either you are completely adapted to the light environment or you're completely adapted to the dark environment, and there's no recombination happening. And so this means that in these situations with gene flow, it's better to have the chromosomal inversion that basically fixes all your locally adapted alleles in a block that isn't broken up by recombination. And so this is why inversions are supposed to contribute to local adaptation and to be favored in such situations. And of course, the results that we find are supporting these predictions from theory that were made in 2006 already. So yeah, when looking at this one contact zone in the snails, what we find is that clearly inversions seem to contribute to local adaptation. So in a separate study, we actually found out that they contribute about 50%. They contribute to about 50% of the phenotypic divergence between the crab and wave ecotype. And we could also find in a separate study that these inversions are clearly associated with specific traits that are different between the crab and wave ecotypes. So we didn't just find that they are highly differentiated between crab and wave, but we could also directly show that they contribute to things like shell length and shell shape. But we do also find scattered SNPs across the genome that also contribute to ecotype divergence. But of course, we have so far looked, we are already 24 minutes in, and so far we have looked only at a single event of divergent adaptation. But in nature, of course, it's in many systems so that you have local adaptation in multiple different geographical locations. So you have similar environmental contrast in many places, and local adaptation happens repeatedly in space. But also local adaptation happens repeatedly in time because environments change over time. So where there's a crab ecotype now might be a wave environment in the future, and maybe the crab ecotype will change to a wave ecotype or the other way around. And so what I'm particularly interested in is understanding, when you look at these different instances of adaptation, is the genetic basis of adaptation always similar, or will it be very different? So how repeatable is the genetic basis of adaptation if you look at different spatial and temporal situations of adaptation? And this is what the rest of my talk will be about. So I'm asking, is the genetic basis of adaptation similar if we look at different geographical scales? So we look at adaptation at different points in space. But I will also, in the end, look at a selection experiment that we did where we followed adaptation in real time. So we can ask, is the genetic basis of adaptation similar if we look at another instance of adaptation in time? And so the Littorina system is a really good system to study repeated adaptation, because these ecotypes that I've shown you before, we don't have them just in a couple of locations, but we have numerous. I mean, these are not all, these are just some randomly placed points, but there will be, in reality, there will be thousands of locations across Europe where you have a crab and a wave ecotype diverging on local shores. So let's start with a small scale. So this will now be a very simple study to understand, because it's basically just a repetition of what I have shown you before about a single contact zone between the crab and wave ecotype. We basically just repeated the same kind of study in 6 more contact zones between crab and wave. So this little island is actually the island where I studied the crab and wave contact zone that I showed you in the beginning. And then we have just 6 more contacts between crab and wave on 3 other islands that we studied as well. And as you can see, this is on a very, very small geographical scale within just a couple of kilometers on the Swedish west coast. And so all the methods we used were essentially the same as I've showed you before. So we use the same kind of hybrid zone analysis separately for each of these different contact zones. And then, of course, we wanted to know, in these different contact zones, do the same genomic regions contribute to adaptation, or is local adaptation having a different genetic basis in these different places? And now we already come to the first results. So what I'm showing you here is when ignoring the inversions, so just looking at the rest of the genome, I'm showing you the proportion of outliers, so of potentially of loci potentially affected by selection, that are shared between the different islands. So and you can, so I'm just showing the sharing between this island and that island, for example, is 39%. And I mean, you can see what it means for the other islands. So you can see that the proportion of sharing is around 40 to 50%. So this is quite a high proportion of outliers that are shared between these different islands, because you need to, I mean, maybe you think, okay, 40%, it doesn't sound that high, but you need to consider that these kind of data, they are always extremely noisy. So even if there was 100% sharing, you would not observe 100% certainly, because there's too much noise in the data. So like 40 or 50% sharing, that is certainly a very high proportion of shared adaptive variation between these different islands. And so it really looks like the genetic basis of divergence is very, very similar on these small geographical scales that we studied here. And then we can look at the inversions. So I'm just showing you the same plot that I've shown you before, where we had just 1 line for the first island. And now I'm just adding the 6 contact zones on the 3 other islands and showing you where the outliers are for those islands. And you can see, I mean, the one thing you can see here, just to repeat that, is that outside the inversions, for example, here, you see that the outliers in the different contact zones cluster in similar regions of the genome, which generates this high sharing that we have seen here. But you can also see that this clustering of outliers in chromosomal inversions, so for example, here on linkage group 6 and 14, is definitely also the case for all these other contact zones that we have studied on the small scale. The exception is here for linkage group 17, which appears to play a role only in the first island that we studied. And then I just quickly made a table where I'm giving you an overview for the inversion. So this one, it will reappear later for the other geographical scales. So here we just have the 7 different contact zones that we studied. Here we have the different inversions that we studied, and I'm just making a cross where the inversion is statistically enriched for outliers in that particular location. And you can see that most of the inversions that show evidence for adaptation do so in more than a single geographical location. For example, this one shows evidence for a contribution to adaptation in 4 different contact zones, but mostly, of course, these 2 inversions stand out on linkage group 6 and linkage group 14 that show evidence for an accumulation of outliers in all the contact zones that we studied. So we can answer the first question: is the genetic basis of adaptation similar in space on small geographical scales? So the answer is yes. So adaptive inversions are shared between these different locations on a small scale, and also a lot of the SNPs are shared, but not all. So there seem to potentially also be some location-specific processes going on. And the reason we think there is so much sharing is, of course, that we are looking at very small scales here. So there is probably quite a lot of gene flow between these different islands. So any kind of locally adaptive allele that appears on one of the islands can be shared with other islands via gene flow between those islands. And then, of course, the interesting question is, does this pattern hold when we look at much larger geographical scales? So here we looked across Europe. So we have samples from 4 countries, 11 locations. So we have Sweden, the UK, France, and Spain. And of course, now we're at a scale where there is a lot less gene flow, because a snail that doesn't disperse very far will have much trouble, coming from Spain to Sweden. So does the pattern change then? So here what we did is we couldn't do this hybrid zone analysis approach that we did in the first part, simply because this is just a larger number of locations, and we sampled a much we sampled a much larger number of locations, and we couldn't do a hybrid zone study in all these places because it's a huge amount of work. So what we did instead is we used a pool-seq approach, where we just sampled crab ecotype snails, and separately we sampled wave ecotype snails, and we pooled all the snails per location and ecotype, and then sequenced as a pool, so we could get allele frequency data for each of the location and each of the ecotypes, but we don't have individual sequencing data in this case. So as I said, we couldn't use the hybrid zone analysis approach here, but we could do a standard genome scan approach, because we have the allele frequency data for all these samples. So I'm not going to go into detail here, but I mean, many of you probably also know this already. But what you do in a genome scan approach is basically you just look across the genome at the differentiation between those samples you're interested in. So here, a crab and a wave ecotype sample from a given location, and you just check where is the differentiation particularly high. And those are regions where selection is counteracting gene flow between the ecotypes. And so these are regions that are potentially affected by divergent selection between the ecotypes. And so again, we have these SNPs or genomic regions that are outliers, which are potentially affected by selection. And so then we can ask, after doing this, to what extent are those outliers that we find for each of the locations shared between those different locations? So this is analogous to what we have seen in the first part, and here are already the results. So this is analogous to this plot that I showed you with all the percentages along the lines. And here we have too many locations to put all the percentages written down, because it would be too many numbers. So what we are showing instead here is we're showing again all the locations, but we're just connecting them by lines, where the thickness and color of the line represents the proportion of sharing of outliers. Again, now focusing outside inversions. So for example, here we have all the Swedish locations, and we can see that they are connected by quite thick dark lines. And this is because they share a relatively high proportion of the outliers. And similarly, if you look at the two Spanish locations, they also still share quite a reasonable number of outliers. But then if you compare geographically distant locations, for example, Spanish and Swedish locations, or at least some of the British and the Swedish locations, then you can see that the lines are thin and there is less sharing of outliers between those geographically distant places. So what we can see here is that if populations are close in space, or locations are close in space, we still have a high sharing of outliers consistent with the first small scale part. But then if we look at large geographical distances, sharing decreases. And then if we look at the inversions, we can again ask which of the inversions are statistically enriched for such outliers, so highly differentiated SNPs between the crab and the wave ecotype. And so that means which inversions are particularly or are potentially affected by divergent selection between ecotypes. And so I'm just adding that to this table here. So here is the part that we've seen before for the small scale, and now I'm just adding the information on the large scale for all the different locations that we have sampled across Europe. And of course, I don't want you to read all these crosses, but the general picture we can see here, I think quite clearly, is that for all the inversions that we found to sometimes play a role on the small geographical scale, we also find them to play a role on the large geographical scale. So many of the inversions seem to play a role in adaptation in multiple different countries across Europe. And in particular, we can see that these inversions that showed the strongest and most consistent pattern in Sweden on the small scale, they also show evidence for selection in all the locations that we studied across Europe. So these really seem to be like globally contributing to local adaptation in the system. And so yeah, what we found in this part is the genetic basis of adaptation similar when we look at large geographical scales. Yes, there is still a sharing of the genetic basis of adaptation, but it decreases with increasing geographical distance. So this is not true for some of the inversions. We find that some inversions are globally shared and globally contribute to local adaptation in the system. And we think a possible explanation for this is that, as I said before, these inversions really, they concentrate adaptive loci in these blocks that cannot be broken up by recombination. And we think, therefore, inversions could also be particularly good transport vehicles for adaptive alleles, because the blocks, they can be transported over large geographical distances without ever being broken up. So that might be a reason why these inversions are globally shared and always contributing to adaptation. And so in the last part, which is something I'm working on at the moment, we actually wanted to study adaptation in the system in real time and also check if we do an adaptation experiment, can we still find the same genomic regions contributing to adaptation that we have found in these studies where we looked at local adaptation that had happened over a long time in different geographical locations in space. And so what we did here, or what we should say Kerstin Johannesson, our collaborator in Sweden, did already a long time ago when I was still very small, is a very nice transplant experiment. So if you look at this map, you can see in this geographical location on the west coast of Sweden, we have an area where we have a wave population. We have an area where we have a crab population. So, okay, there's nothing particularly exciting there. But we also have this very, very small island, which is called a skerry, which you almost can't see on this satellite image because it's so small, which you can also see in this picture. So it's almost not an island. It's really just a rock. And the interesting thing is that in 1988, all the snails that used to live on that rock, they died because there was a toxic algal bloom. So the island was completely empty of snails. And as you can see here already, this island is a wave habitat because it's in the middle of the sea, it's exposed to waves, and crabs cannot live there. so wave snails had lived there, but they were all dead because of this algal bloom. And then Kerstin thought, okay, we have a wave habitat here. What if we take some crab snails, place them in the wave habitat, and see if they evolve into wave snails again? So that's what she did. She took snails from this crab population in 1992 and placed them into this wave habitat. And since then, we have been looking at, or she has been looking at for most of the time, what is happening on that skerry out in the sea. And so we could look at the phenotypic change on that skerry. And one thing that I forgot to say is that if we see adaptation here, we should note that there are two possible sources of adaptive alleles. So one source is standing genetic variation that the crab population that Kerstin placed here already carried with it from variation that was present in the crab donor population. So in that original crab population, the crab individuals were taken from, but there's an additional source of adaptive variation or potential source of adaptive variation, which is gene flow from this nearby wave population. So at the moment, if there is adaptation, we cannot say if it is due to gene flow or due to standing genetic variation from here. And so if we observe the skerry population over time, we can see that indeed it has evolved from this crab ecotype like snail towards a snail that almost looks like, or it looks more or less perfectly like the wave ecotype. So this is just a model of the shell shape and size. And each of these models represents the average of the snails sampled at that respective time point. So you can really see a very substantial phenotypic change towards the wave ecotype. And you can also see that the change is almost done already by the year 2005. So after about 13 years, the snails the population has more or less evolved into the wave ecotype. And so of course, we also wanted to study this genetically. So at different time points, we sampled at 3 different time points, 2005, 2018, and 2021. We sampled individuals from this population, and we genotyped the population for a set of SNPs. So we included some neutral SNPs that we expected to do nothing particularly very interesting. So we didn't expect them to be under selection based on all the information we had from all the other locations that we have studied. But we also included some outlier SNPs. And it is really important to note here that outlier SNPs are not outlier SNPs anywhere in the system necessarily, but they are SNPs that we took from the previous studies. So we took some of the studies that showed evidence for selection in Sweden in this small scale study or in this large scale study. And we included them here. So basically we could ask those SNPs that we found to be under selection between crab and wave in previous studies, do they also respond to selection in this experimental population? And of course, that's why we included them. And we also finally, we included some inversion SNPs so that we can also see the frequencies of the inversion arrangements changing in the skerry population. This I already said. So now here I'm just showing you a couple of results from this study. So first I will show you some neutral SNPs, how they changed over time. So we have here, we have on the x-axis, we have time. So I'm not labeling the x-axis because I wrote down the years here. So we are just looking at these different years that we sampled on the skerry in this bright green color. And there we see how the allele frequency changed for these 8 example SNPs. Again, of course, I am not showing you all the SNPs. And for comparison, we have the allele frequency for the same SNPs in the wave population. And you can see here that if you look at these putatively neutral SNPs, some of them are changing, some of them are going up in frequency a bit, but overall, you cannot really see that there is a lot of change for these neutral SNPs. But then if we compare, can we do it? If we compare to the outlier SNPs from previous studies, you can see, okay, some of them are also not changing very much, but some of them are changing quite a lot over time on the skerry. And so the overall picture is that for the outliers, we have a lot more change on the scary than for the neutral SNPs, which is consistent with there being selection on the outlier SNPs on the scary, as we might have expected. And so what we can do is we can, it's a bit similar to what I talked about for the first study. We can also here establish a neutral model of allele frequency change that we expect if there is no selection on the scary. And this neutral model is reflected by these blue lines here. So here what I'm showing is simulated changes of allele frequencies under neutrality over time from the start to the end of the experiment. Yes, if there was no selection, right? So this is what we see in blue, but in green, we see, again, one of those outlier SNPs, how it changes actually on the scary. So that is what we actually observe. You can see that this SNP is changing much more than what we would expect under neutrality. And so we can infer that this SNP is likely affected by selection on the scary. And overall, we find that 13% of the outlier SNPs change more than expected under neutrality, so are potentially under selection. So obviously, this is not the majority. So many of the known outliers do not respond to selection here on the island, apparently, but 13% do, and some obviously quite clearly. And then finally, we can do the same for, or something similar for the inversions. So I'm showing similar plots for the inversions here with the neutral expected change shown in blue and the observed change shown in green. And every plot is a different inversion. And you can see that at least for some of the inversions, so particularly for this one, and this one, we can quite clearly see that the change is not consistent with neutrality. So we seem to have really rapid adaptation here within just a couple of decades based on these known inversions and probably also some of the outliers that we have detected in previous studies. So if we finally look at this big table again, we can see that some of the, I mean, this is what I already said more or less. We can see that some of the inversions that we know from previous studies also contribute to adaptation in this experiment. And maybe most interestingly, those inversions that have shown a pattern in all these locations that we have studied, they also very strongly respond to selection in this experiment. So this is this inversion and this inversion that shows an extremely strong pattern of going from almost 0 to almost 1 in the course of just 30-year experiment. So these seem to really strongly contribute to adaptation in this system. So yeah, we can, if we want to answer our question, is the genetic basis of adaptation similar also in time if we do an artificial selection experiment? And we can say yes, to some extent it is similar. We find some of the outliers that we have detected in previous studies also contribute to adaptation here, and certainly some of the inversions do as well. But we also find that not all the known outliers contribute to adaptation. So it seems like actually potentially some subset of inversions and outliers is enough to generate the full phenotypic adaptation that is necessary on that little island to withstand wave action. And with this, I would like to finish. So just quickly, a few take-home messages. So we have clearly identified very strong candidate regions for adaptive divergence in this system. I mean, I think these two inversions that show patterns in all the locations and the experiment, I think nobody can say they don't contribute to adaptation. So I think the evidence there is very strong. We also find that the genetic basis of adaptation is shared over space and time, and particularly on small geographical scales. As I've said many times, we find that inversions play a large role in adaptation in this system, and even on very large geographical scales, because they seem to be shared by gene flow across many different locations. And finally, we also showed that based on this adaptation, based on this adaptive variation that we have studied in previous work, we can also see adaptation in a selection experiment where we have very, very rapid adaptation to a new environment, which is probably mostly based on rapid changes in inversion frequencies. And with this, I'm finishing. So I would like to thank a lot of people that were involved in the work that I've shown. For example, Roger and Kerstin, the PIs in Sheffield and Gothenburg, who have done a lot of this work and overseen a lot of this work that I did during my postdoc. And I would also like to thank Diego, who is my PhD student at ISD, who is working on the SCARY system at the moment. And then, of course, I would also very much thank you again for the invitation and for listening to my talk today. And I'm very happy to answer questions.